So today we're joined by James Higham, who is a newly appointed full professor in the Department of Anthropology at New York University. Professor Higham is an evolutionary anthropologist and primatologist with a very broad range of research in his portfolio, drawing from both observational and experimental studies of non-human primates. His work spans the scales from molecular to demographic, including both functional and physiological analyses of behavior, structural morphology, aging, and life history. So thank you very much for joining us today, James. We're all very excited to learn more about your work. James' talk today is entitled Insights into Life History from the Cayo Santiago Rhesus Macaques. Welcome to Beck.
Thank you. Thanks very much. And thank you so much for the invitation. Wish I could have made it over in person, but unfortunately it wasn't quite on the cards this time. Sorry, just to be clear, do I speak for an hour or 45 minutes or 50 minutes or whatever? Okay, all right, well, let's see how.
All right, any space between 45 and an hour is fine.
Okay, that sounds great. All right, thanks. So before I start, hopefully these slides are moving over for you. Before I start, I just wanted to thank a number of people, including the Caribbean Primate Research Center, part of University of Puerto Rico, my host institution, NYU, and especially to NIH and to NSF and the Leakey Foundation. There's other funders that have funded our work, but these three have been particularly consistently generous and supportive. And a long list of important collaborators, including my co-PIs on a number of the work that's funded by the NIH, primarily Lauren Brent and Noah Snyder-Mackler. So my own interests were, as Brian said, were always kind of broad, and I sort of saw myself as sitting at the intersection of social behavior and social biology and cognition and the brain and decision making and communication and sensory ecology, and exploring those interactions. And the thing that always really struck me was the amount of variation and diversity that we see both within and between species. And so I was always interested really in trying to understand both intra and interspecific variation, where it comes from, and how exploring really this intersection. And another way of looking at that is that I've always been interested in exploring and interrogating this kind of interface where the biological meets the social. And I try to do it from both sides, really, like a bottom-up, if you like, perspective of asking questions about how variation in the genome and the epigenome via the transcriptome build cellular phenotypes, which scale up to tissue and organ level responses in stimuli, and how those tissue level processes structure decision-making at the levels of individuals, and in turn, how those individual level decisions and behavior structure societies and populations. And also from this top-down perspective. And I think anthropology is kind of like particularly good at getting at this relative to other components of the biological sciences. Like, how do the behaviors and structures of populations and societies get under the skin? How do they impact individual health, tissues, organs, cellular phenotypes, and structure patterns of transcriptomic expression and the epigenome? I was always personally very interested in sexual selection. And for those reasons, I started studying African and Asian monkeys. The primates on the whole have a really interesting, we come from a really, really interesting order, which has incredible social and mating system diversity. Every kind of social and mating system that you can imagine is exhibited within our order, including relatively rarely for large terrestrial mammals, polygynandrous mating systems with multiple males and multiple females. And interestingly, in the primates really divergent outcomes in what that looks like. In some species and some populations, alpha males are siring 90–95% of sires in the group, whereas in others it's very flat. It's a flat reproductive skew, and there's not much variation in what males are doing according to their social status. Females in anthropoid primates exhibit proceptivity and receptivity that's extended outside of fertile phases, and that extends the period over which males must compete for female fertile phases. And a big outcome of all this variation is a lot of very interesting traits, actually, that are not really seen in other types of clades. So a mix of traits that are related to mutual mate choice and mating competition. So on the whole, the large terrestrial mammals are not a very colorful bunch. They tend to exhibit background matching, fur that's covered all over and that match their backgrounds, whether that's a gray or a white or something else. They exhibit counter shading, being darker on their backs and lighter on their bellies, but mainly what they are is weaponized. Females, so the argument goes, are easy to kind of aggregate. They aggregate in order to reduce their predation risk, and then it's easy for males to try to monopolize them, and so there's selection on weaponry, and we get the evolution of antlers and horns and large canine teeth and tusks and all kinds of weaponry. And so the argument goes that really in large terrestrial mammals on the whole, the predominant major sexual selection mechanism that's operating is male-male competition. Whereas in some of the other clades, like birds and fish, some of the groups of fish, they tend, instead of being weaponized, to be highly ornamented with crests and trains and feathers, very colorful patches and things. And here it's lots of intersexual selection and female mate choice. So we have these very brightly colored males, and they prance around in front of the females, who then pick between males on the basis of their ornaments. So the classical kind of arguments go that the large terrestrial mammals evolved their armaments, and the birds and the fish and other clades evolved their ornaments. And then we have the primates, in which we have this really interesting mix of very colorful, of course, primates have evolved trichromacy in a number of groups, including uniform trichromacy across the catarrhines. That's monkeys in Africa and Asia and the apes. But a real mix of traits. So here you've got two male drills squaring off opposite each other. They look absolutely magnificent. These huge cheek flanges, these paracolossal bones, this red under the lower lip. And this drab little thing here is a full-grown adult multiparous female. And so males have evolved 3 times the mass of the females. And so we have these interesting mix of traits, mutual mate choice and mating competition. Of course, primates are highly social. They have individual recognition and repeated interactions, and they develop social knowledge about each other, in which the signals and the behaviors that they exhibit and also interpret from each other are richly placed into this rich backdrop of social knowledge, in which individuals recognize things about each other, or at least treat stimuli from each other differently depending on their prior experience and interactions. And another thing about primates that makes them a really great system, if you're interested in questions of variation, diversity, communication, cognition, which is that they have a really highly radiational evolution, in which lots of species have been produced that are relatively closely related to each other in relatively short amounts of time for such large animals. So it's thought that we have ended up with 4 different genera of gibbons, which are extremely slow life history, all within about a period of 50,000 years, which is extraordinary. And you can see all of these species of guenons. They're a clade of monkeys from West and Central Africa primarily. But actually their skulls look very, very similar. A lot of this variation is soft tissues in different colorful patches and ear tufts and crests and white spots on their noses and things. So in some of my other talks that I give, I talk about these components of my work, thinking about radiational evolution and the production of lots of species. But I thought for today, I really wanted to dig in to one of what's become really kind of like my biggest investment in time in terms of field sites and study systems, which is this little island off the coast of southeast Puerto Rico called Cayo Santiago. And Cayo Santiago hosts a population of macaques. Here's an old photo of Cayo Santiago from the 1930s. And what happened was a guy called Clarence Ray Carpenter, who was a forerunner of primatology in North America, went over to India, trapped lots of macaques, brought them back by boat. He looked after them himself. He sold a few when he got back to the mainland, and then he ended up releasing 409 of them on this island. And his idea really was that it would be a sort of natural laboratory, part field site, part lab, in which he could do experiments. He could observe them as they free roamed and see what they did, but also that he could trap them and measure them and do experiments with them. And this actually is a classic photo of one of his assistants doing an experiment. Actually, what's happening here is that they've separated mothers from the infants, and then they put the infants on a little platform over here and then open, and they let the mums run out and grab them. So they were doing things like maternal separations and things. So it really was an idea to be observational and experimental and be part field and part lab. And I hope that I can show you in the talk that his vision for this has been achieved. We have vast amounts of data types. So that was in 1938. And after the Second World War broke out, Cayo Santiago was abandoned and there was no research going on. And it was actually Stuart Altman who reestablished research there in the 1950s. He went to Cayo Santiago in 1956 and was there for a couple of years. And he established a life history and demography database. He started doing a daily census, tattooed the animals for individual recognition. He started putting that together in 1956. We take life history and demographic records from about 1958 with a little bit of burn-in for the initial very patchy years. And even in 1958, it's patchy, but it gets better and better, as you might imagine. There's currently around 15 to 1800 monkeys on Cayo Santiago from 409 founders, and it's not an inbred population. They've seemed to have outbred themselves by dissociative mating, which is interesting. The genetic pedigree started in the early 90s, but from individuals that were already kind of like 6 or 7. So that from individuals born in about 1985, we have a full genetic pedigree of everyone. The skeletal collection was started in 1971. And we have thousands of skeletons as a load here now at NYU from more recent individuals, and then anything from 2010 and earlier is in Rio Piedras at the Medical Science Campus of the University of Puerto Rico. We've been building up extensive biobanks of functional omics data, blood, tissues, massive microbiome data sets. It's been undergoing for like the last since 2016, the Caribbean Primate Research Center have been removing and humanely euthanizing while trying to experiment with contraception, population reduction. Those aren't our decisions on which animals get euthanized, but we have been trying to maximize the research advantage of that by biobanking individuals. So once they are taken to Savannah Seco, we collect microbiome from every area, we measure their soft tissues, we do eye exams and various other things, and then post-euthanasia, all the tissues are collected, biobanks for omics work, we keep the skeletons, the sensory organs, we go up the gut and in the lungs and do the microbiome gut section by section. So we really have built some very large biobanks coming out of Cayo Santiago. There's really big behavioral databases, lots of fecal and urine sampling over the years. We're also able to trap and release the same animals year after year, and we can collect blood samples, morphometrics, joint mobility, all kinds of things. So really, there's a lot of scope, many different kinds of data types emerging from Cayo Santiago. And we're also able to investigate things with a lot of depth. And so what I wanted to do in this talk was start by giving you an example of a phenotype that we've been able to explore with a lot of depth using this field site, and then go on to show you some of the scope and some of the range of different kinds of data types we've been collecting and how we're starting to integrate them. So first, I wanted to choose a trait that we've done a lot of studies on and I could really pick apart for you. And so I thought I would do this looking at going back to Tinbergen and Tinbergen's 4 levels of explanation, looking at the mechanism of a trait, looking at the function of a trait, looking at the ontogeny, so the development of a trait. I'm not going to talk about evolution and phylogeny of this trait here, because of course that requires comparing between species, and I'm going to focus on Rhesus macaques. And the trait that I've chosen to talk about is a trait that we've published a lot of studies on, and it's this bright red to pale pink facial coloration, which is exhibited by both sexes. So this is a female, relatively pale pink face. This is a female, much darker face. This is a male, dark red face. This is a male, pale pink face. And as I said, I'm going to talk about the mechanisms underlying this, the function and the ontogeny, and the evolution and phylogeny part. For that, really, we study other species of macaques, we study other species of baboons, and we try to build comparative models of some of the comparative variation that we see. So this is an interesting trait, I think, for multiple reasons, which I'll dig into. But one thing that should strike you straight away, we have thousands, more than 10,000, maybe 15,000 plus good standardized images of Rhesus face and genitals and rump areas, standardized for color. And if you plot the males and females on top of each other, they overlap fully. So you would think that this would be a sexually selected trait, sort of instinctively, and there's a lot that seems to be going on with this trait, but most sexually selected traits are dimorphic. They're not fully overlapping in their phenotypic exhibition. And Darwin was very interested in these kinds of colors of monkeys. He wrote a paper in Nature in 1876 that's simply called On Sexual Selection in Monkeys. And he wrote that the discussion of sexual selection in My Descent of Man, no case interested and perplexed me so much as the brightly colored hinder ends and adjoining parts of certain monkeys. And these kinds of monkey sex skins, as they're sometimes called, are really interesting. Darwin also wrote about the red faces of Rhesus macaques specifically, and expressions of expressions and emotions of man and other animals. The interesting thing about this kind of trait is that Rhesus macaques are in a very unusual dynamic for a large terrestrial mammal, and even within the macaques, they're pretty unusual, actually. Most African and Asian monkeys are either in polygynous or polygynandrous mating systems, where they have direct competition over females and indirect female mate choice. And so this is a crested macaque. They have these massive canines. Males are 2/3 larger than females, about 1.65 sexual dimorphism ratio. And they have direct contest competition. The alpha male comes in from the outside, he attacks the resident alpha male, and if he's successful, he takes over the group and he commits infanticide to the infants and stuff. And as a consequence, strong body and canine sexual dimorphism. Rhesus are in a really interesting dynamic where the body and canine size dimorphism seems to have come down, that males are only about 30% to 35% larger than females. And the canine size, the canine dimorphism is not so great, but look at these testes. absolutely massive testes, and you compare that to the crested macaques, these tiny little testes over here, you can see that we're in a dynamic here of reduced direct contest competition and it increased scramble competition, sperm competition, endurance rivalry, these kinds of other mechanisms. And that doesn't mean that there's no direct competition in Rhesus. There is. They're still sexually dimorphic in canine and body size. But this is an interesting dynamic. And what males seem to do is they turn up on the edges of groups and rise by a process of succession as higher ranked individuals leave or die. And so we've studied colorful traits in species like this crested macaque or in drills, and they've been studied in gelada by Tori Bergman and Jacinta Bina. And really, it turns out that these colorful patches in a lot of these primate species are actually not as interesting as you might think, in that they're also just a product of male-male competition. They signal male-male social status, and there's very little evidence that they're actually ornaments that are attractive to females. But over here in Rhesus, we see something quite different. So I'm going to dig into this a little bit. We take fecal samples and urine samples, and we measure and model signals. And because Rhesus have such well-characterized vision, we can model their vision in terms of retinal sensitivity for Rhesus macaques, how stimulated their retinal receptors would be, and look at their color discrimination thresholds. And we can see that for females, the mechanism is that across the cycle, there's very little change in actual color, but the faces darken. And just before ovulation, where 0 is ovulation, the faces darken relative to a few days earlier. So this is the luminance getting darker. These are perceptual units, just noticeable differences, where each unit is one discrimination unit within the visual system of a Rhesus monkey. For males, it's pretty interesting because so what we know is happening here is that estrogen is rising during the follicular phase of the cycle. It causes a darkening of the facial skin, and then it lightens. After ovulation, you get the corpus luteum, estrogen goes down, progesterone goes up, and the face lightens. In males, in theory, it should be related to testosterone. If you inject a male with testosterone, they flush. But actually, there's a range of data from a range of species now showing that there's a dissociation between testosterone concentrations and the reddening of the skin, actually. So in gelada, for example, the males get these testosterone rises. The bachelor males, so you have these one male units, you have a one male unit holder, and he has a bright red chest patch, and multiple females. And then you have these bachelor males who all have pale pink chests. And if you inject any of those males with testosterone, they go bright red. But in natural circumstances, their testosterones go up before they challenge the resident holder, and their chest patch is still pale pink. And they get really aggressive, and then they challenge. And if they win, only if they win, then they go bright red. And the deposed male then goes pale pink. So our idea about this is that it's perhaps related to the receptor expression, which is also changing in the skin. And we're lucky in Cayo Santiago because we can actually take tissue punches and look at tissue-specific gene expression of the estrogen receptors, of the androgen receptor, of the progesterone receptor. Because we have a full pedigree, we can also look not only at hormonal mechanisms, but we can ask questions about how much genetic variance there is that's being contributed, and there's nice heritable signals, especially if you look at same-sex parents. Male coloration has a higher additive genetic variation if you only look at the paternal line, and females if you only look at the maternal line, probably because some of the apparatus like the sex steroids that are involved, you're also inheriting receptor components associated with that from your same-sex parent. We can test the function of these. So we can look at the mechanisms, we can look at the underlying genetics, we can look at gene expression, we can look at the hormones. We can go and test the function using experimental approaches. These are approaches that we sort of stole from developmental and comparative psychology, where you have a presenter who shows two images of different individuals, one with a red face, one with a pale pink face, and then there's a video, over here, one with a red face, one with a pale pink face, and there's a video person here recording the monkey's responses, and then those videos are coded frame by frame. And if you look at female coloration, males prefer looking at ovulatory faced females over over pre-ovulatory faced females, especially, and this is driven by these experiments were run in group V on Cayo Santiago, and the effect is really driven by males who are familiar with the females in the images, that are really picking their signal variation and are particularly interested in those females. And this kind of effect is really interesting to explore because it's that kind of social knowledge and familiarity component interacting with communication. We can also undertake behavioral observations, and we can show that not just within females experimentally, but between females, males prefer redder face females. So this is number of mating series. They prefer redder face females and they prefer dark face females. So dark red face females are what males prefer. And we can use quantitative genetics and calculate selection gradients on these traits. And we can show that there is linear positive selection on females' redness. Females who have redder faces on Cayo Santiago have been having greater annual fecundity over time. So of course, males that are selecting for red-faced females will be doing better because these females have higher fecundity. That, of course, could be just related to their underlying health. Females might have more oxygenated blood, better blood flow if they're in better health, and they might be the females that have higher fecundity. But nonetheless, if males are choosing those females and choosing females based on that trait, they'll be doing better. In males, and I think this is a really interesting result, females prefer darker red-faced males. So this is number of female solicitations according to male coloration, with the dark red-faced males getting the most solicitations observationally. And we've backed that up experimentally. So we've done these looking time experiments. You record frame by frame interest in the different images, and you have a nice matched design. And females particularly, but not males, are the ones that look more at dark red face males, especially driven by young females in this particular experiment, and we've replicated it since. So females are interested in males based on their coloration. This is unusual, actually. In a lot of these other, I think people tend to think that perhaps the red faces of Rhesus are analogous to the red on a mandrel's nose or the red chest patch of a gelada. But as I said, those signals are really related to male social status. It's not related to male social status in Cayo Santiago. It's an ornament. The females are choosing between males based on the color of their faces. And that's very unusual for a large terrestrial mammal. Even for a primate, that's very unusual. And this is more like a trait that you might see in a bird, like those birds of paradise I was talking about earlier. And as I said, it's not related to social status. Male coloration and the face, the rump, the genital is not related to dominance rank. We've replicated this across years, across different data sets collected by different people. There is some evidence it might be secondarily selected to have a role in male-male competition. There seems to be a slightly increased likelihood of aggression when males are more similar in coloration than when they're more distant in coloration. So some evidence for some male-male competition. And we can also undertake selection gradients on male coloration. And what we find is that the males who've had most offspring, the highest fecundity on Cayo Santiago over time, are those combining having dark red faces with high dominance rank. If you have a high dominance rank and a dark red face, you have the highest fecundity, and those with the highest fecundity up here, and those with the lowest fecundity over time are having pale pink faces combined with low dominance rank. That's tested with the correlational selection model. It's not really correlational selection because dominance rank is not a heritable trait. It's something that they acquire during their lifetime. But it's tested with a correlational selection model, but I personally would not call that correlational selection. But clearly selection acting on male coloration. So what we've got here is a really interesting trait that's varying within individuals, between individuals, is being used in mutual mate choice by both sexes independently. And so you're ending up with these really complicated fitness landscapes, really complicated selection acting on the trait. It's really interesting. And of course, because we track individuals across the lifespan, we can look at ontogeny, we can look at how the redness and the luminance, the darkness of the trait change over time. And we can also test experimentally and observationally who's interested in this. And we can show, for example, that if you, in this experiment, actually juveniles look most at the faces, but they don't discriminate. Once you get to adulthood, they're interested in more looking at the dark red faces than they are the pale pink faces. And it's not that juveniles aren't interested in looking. In fact, they're more interested in looking. Their overall looking time is greater, but they don't care. They're just looking for social information. And once they hit sexual maturity, then you see these differences start to emerge in their salience and what they're really attending to. So we can attract not only ontogeny of the development of the trait, but ontogeny of interest in the trait in other individuals. And so what I hope to do in this kind of first big part of the talk was just show you how, with a long-running field study in which you have a lot of capacity like this, we can really go into phenotypes and dig into them with study after study. We can look at the ontogeny of them. We can test the effect that they have on the behavior of others. We can look at behavior. We can test it experimentally. We can get hold of the skin itself and look at patterns of gene expression in the skin. We can look at the underlying hormones from feces and urine. We can really dig into a trait. And this is now, a sexually selected trait where we can do quantitative genetics. We can look for selection on the traits. We can really dig in and investigate traits with a lot of depth. So I want to switch to kind of like the second main part of my talk, which is, yes, we can do all of that depth, but we also have huge amounts of scope with that kind of field site. So this is a figure from one of our NIH grants in which we're using observational data to look both cross-sectionally, ignore the sample sizes because they're for a specific grant, cross-sectionally across hundreds of individuals, as well as longitudinally at change over time. We can measure lots of biomarkers. We can measure things in urine and feces and blood, immune and physiological markers. We can measure physical things like body condition, range of motion, facial aging, muscle mass, gait speed, and other kinetics. We can look within blood at DNA methylation, telomere attrition, mitochondrial function, protein stability, and build really integrated analyses of what's happening across the lifespan. So, as I said, we have these big longitudinal and demographic databases. We can look at female fertility across the lifespan. We have this early period when females are around 3 or 4 in which their fertility is low, and then they get into this nice prime phase in which their fertility is sustained and quite high. And then we can see these clear aging effects on female fertility. We can also see independently to an aging effect, a terminal effect that's age independent. However old you are, whether you're pre-prime, whether you're prime, whether you're post-prime, you have a reduced level of fertility in the last year or the last couple of years before your death. And that's age independent. That happens when you're young, whether you're old. And we're probably detecting these frailty and disease conditions coming in. And we can dig into that with, this is the result of a hidden Markov model in which we are identifying hidden states. And so females are in a reproducing state, and they cycle back and forth into a non-reproducing state. This is the probability of them going from one state to the other once they've reproduced, the probability of not reproducing and then going back into reproducing again. Sorry, this is the probability of them staying in the hidden state 1.69, and then there's also a probability for those that transfer the probability of them doing it in a given year. And we can identify this, those models identify a hidden state, a frailty state, which we called in this paper inescapable terminal decline. And the reason for that is that it's inescapable in that once you're in this hidden frailty state, you largely don't reproduce. There's a small chance of reproducing, but you largely don't reproduce. It's terminal in the sense that it only leads to death, and it's inescapable. You can't go back. So there's this inescapable terminal hidden decline state, a kind of frailty state that we can identify with these kinds of models. And this is the entire life. These data are longitudinal. They're the entire life history of individual females, hundreds of them, from birth all the way to death. This is not a cross-sectional analysis in which we can track individuals and exactly what happened across their lifespan. And we can see in these analyses, females going back and forth between reproducing and not reproducing as they age. And then this hidden state, the frailty state, really coming in as they age. It really kicks off after around 17, and then after they're 20, it really kicks up. And you can really use these models to interrogate the aging process. We can also track infant mortality across the maternal lifespan. This is an analysis we published in PNAS a few years ago, showing this really nice relationship between short interbirth intervals here and elevated risk of infant mortality. When you have short interbirth intervals, you have higher infant mortality. And the interesting thing about that is it's a quadratic, actually. Long interbirth intervals are also bad. This is a pattern that was identified in humans and thought by many to be a social cultural pattern, actually, and to do with changes in human lifestyles and things. But actually it's here in the macaques too. We're not really sure why these long intervals also lead to high infant mortality. Perhaps we're picking up on some characteristics about females themselves. And these females are females in poor condition that are having high infant mortality and long interbirth intervals. But these are the kinds of insights into life history you can get from these huge demographic databases, insights into human life history too. And just to say, like, these interbirth intervals are based on the births of over 9,000 offspring from over 1,400 mothers. So we're able to get these really, I mean, for primatology, these are massive data sets, and we're able to look at these kinds of aging effects across the maternal lifespan. And we can integrate our different data types. So we weigh individuals, and so we have data on infant mass, and we can show that all females give birth to infants that have low body weights, and that infants with low body weights have a higher risk of death. For every 100 grams, they have a 100-gram difference. They have a 1% increase in their chance of being dead before they're 12 months. And we can calculate this based on hundreds and hundreds of infants, infant masses. So this is the relative hazard of death and deviation from mean infant body mass. So if they're large, they have a reduced, if they're heavy, they have a reduced hazard of death. And if they're light, they have an increased hazard of death. We can look at the microbiome across the lifespan and integrate that with other measures. The rectal and oral microbiomes are relatively similar between infants and adults. As you might expect, the infant and young vaginal and penile microbiomes are very different than adults once they start having sex in the population. We see big changes. We can look at soft tissues and skeletal development across the lifespan. This is body mass, ontogenetic curves, growth rates. we're able to integrate those kinds of measurements with things like joint mobility across the lifespan, and gait speed, and your ability to do to get around and interact with others. I find this pretty depressing, actually. This is by 15, all individuals have lost kind of 10 to 15% of their hip extension that they had in the first couple of years of their life. I'm going to talk about hurricane effects a little bit later on. Because of all this biobanking that's been going on, we have the brains, and we've been able to look region by region across the brain at signatures of aging cross-sectionally. Of course, not longitudinally, we can't measure them point by point. And you can see that there's genes, this is a gene, for example, that increases across lots of different brain regions by age, whether that's, V1, like the visual cortex, or something like the LGN. And also we can sort by cell types, and we can look at cell types like oligodendrocytes, which are increasing by age, region by region, tissue-specific gene expression. We can look at sensory systems across the lifespan. We have detailed eye exams that we've been taking, OCT, fundus images. These are cataracts. This is drusen, little bits of salt and protein accumulating in the eye that will affect the vision. These are all important for vision. Your spherical equivalence is related to increased astigmatism, axial length, increases issues with myopia. And we can look at behavior across the lifespan. This is in prep work showing that the number of grooming partners and proximity partners declines within an individual as they age over a decade or more. And the interesting thing about it is it's them driving it. They approach fewer partners as they get older. It's not that other partners don't approach them. They approach fewer partners and are more socially selective in who they invest their time in. And these are effects that we see in humans too. Sorry, I keep using my cursor to point at the wrong screen. So grooming partners declining with time, proximity partners declining as they age. And it's driven by them. The number of partners that they approach is reduced. So increased social selectivity. We see these patterns in humans too. And of course, we can start to package all of this together. Like if your mobility is declining, if your sensory systems are declining, if you're cognitively declining, your interest perhaps in interacting with others may decline too, because you're getting less out of the interactions. It's more difficult to walk over there as your hips stiffen. In the very old individuals, they have fused vertebrae. You can see it. They're walking very, very stiffly. They're very stiff back and stiff hipped. So you can really see across the lifespan in brains, in microbiome, in social behavior, in sensory systems, in changes. You can track everything. It's hugely integrative. And by the way, just to say, if you're interested in social aging, we just had a review out in Biology Letters recently, written by an excellent postdoc, Erin Siracusa, on social aging and why individuals may exhibit late life changes in social behavior and different kinds of explanations for it. And we are just starting to get to the stage where we're integrating across these massive kinds of data sets, in which we're integrating our social behavior with gut microbiome, exploring that gut-brain inflammation axis. And we're finding lots of interesting things already. So this is a figure that shows pathways that are related to immune responses and signaling pathways that are all upregulated in the brain in patterns of gene expression in relation to the number of social partners and in interaction of the social partners and the microbiome in the gut. So there's really exciting things, I think, that this integrative kind of biology approach is headed. So I showed you some of the depth that we can get into by looking at one phenotypic trait. I've shown you some of the scope and the scale of what we're able to do on Cayo Santiago. And I just wanted to finish a little bit by talking about Cayo Santiago and the Anthropocene, because a lot has been going on. Of course, we're all aware of the SARS-CoV-2 COVID pandemic. And we were very quick, we had a preprint up looking at primate predicted risk and susceptibility to it. In March 2020, we were quick to get this out. And we were able to show quite quickly that the amino acid residues for ACE2, which is the primary viral target receptor for SARS-CoV-2, were in fact uniform across all catarrhines, across monkeys in Africa and Asia and apes. And they're all therefore predicted to be very susceptible to the disease in the same way that humans are too. And in that paper, we were also able to undertake computational protein-protein interaction modeling between the target viral receptor on our epithelial cells and the virus, and look at how individual amino acid changes in sequences are predicted to affect binding affinity of the virus for different species of primate. And it quickly became apparent that Rhesus macaques are an excellent model for COVID research, and that has had quite quick consequences. This is an article from The Atlantic. America is running low on a crucial resource for COVID-19 vaccines. The country is facing a monkey shortage. If you've had a COVID vaccine, it will have been developed through testing on Rhesus macaques. The Rhesus macaques that are used for that kind of vaccine testing are SPF monkeys, animals that are kept in a specific pathogen-free colony and are free from some macaque viruses. But increasingly, there's pressure on even on conventional animals that have, for example, simian herpes B virus. And that's starting to affect the Sabana Seca facility in San Juan. And it may be that monkeys that are removed in future for Cayo Santiago end up going into scientific research rather than into biobanks or anything like that. But of course, those are decisions that we as researchers are not making. We don't manage the population. That's the Caribbean Primate Research Center. One of the biggest things that's happened on Cayo Santiago in a long time is this Hurricane Maria, which went straight over Cayo. Cayo is a tiny little dot just here off the coast of Puerto Rico. All of Puerto Rico, you can see, is completely covered by Hurricane Maria. And it hit Cayo before it hit mainland Puerto Rico and destroyed every building on the island, most of the vegetation, and led to a swathe of new studies on the top-down environmental impacts of climate change and natural disaster on our study subjects. So this is a big piece in the New York Times if you'd like to go and read it. Primal Fear: Can Monkeys Help Unlock the Secrets of Trauma? And you can see them here, all this, there's some vegetation growing back, but a lot of dead vegetation just lying around and a largely devegetated environment. And we've just started to get our first papers out looking at the effects of this hurricane on the population. Here's a recent one. It's on the cover of Current Biology. You can see all this dead vegetation and the animals scrounging around afterwards. And interestingly, we showed, and this is work led by an excellent PhD student, Camille Testar, who showed that social networks increased and that Rhesus macaques built new social connections in response to this natural disaster. And it was particularly individuals that were isolated and socially isolated who went out and sought out and were proactive about building new social relationships. And so we can see this is a greenery index. You can see this massive decrease in vegetation when the hurricane struck in September 2017. This is mortality on the right in figure B. You can see a little spike in post-2017, post-hurricane mortality, not a huge one, not as big as the Shigella outbreak they had there in 2010, for example, but a little increase. And then you can see increases in proximity networks and grooming networks in 2 different groups and different parts of the island that we were measuring at the time in response to the hurricane. And remarkably, we just published this in PNAS. This is again over here, big differences in greenery, devegetated environment. If you look in the blood at transcriptomic patterns that are related to age, so here is an age signature, PC3, patterns of gene expression. Older individuals have a lower load lower on PC3. For a given age, individuals that lived through the hurricane have a lower score on PC3 than individuals that did not. So they're essentially molecularly older for their chronological age than individuals that didn't live through the hurricane. So coming back to this biosocial interface, these kinds of studies, we're able to see these top-down effects. We're able to go bottom up and explore this biological and social interface, genotypes to phenotypes, up into population, how things like whether it's individual decision-making on who to mate with on the basis of their color or other attributes, how that builds and structures populations and affects the demography of the population over time, relates to fecundity. But we're also able to really interestingly do this top-down stuff. I showed you, for example, those relationships between age. There's huge amounts of scatter in the relationship between a lot of these variables and age. Where's that coming from? Some people get cardiovascular disease at 40 and other people will never develop it in their life. And some of that is genetics and some of it is not. The social environment and social support is a top predictor of health. It's one of the better predictors of 5-year cancer survival rates, for example. How does variation in your social environment, your social status, your degree of social support, how does it get under the skin and affect your health, your biology? How do things like natural disasters, broader environmental senses, broader environmental conditions get under the skin, that top-down component of this biological and social interface? So with Cayo, we're able to really pick this apart. And I just want to finish with a quote from Sherwood Washburn, who was one of the forerunners of biological anthropology as it is today. He wrote a paper in the early 1970s called The Promise of Primatology. And he wrote that the promise of primatology is a fundamental challenge to the notion that the social should be studied without reference to the biological. It's something that Nate Dominique and I recently revisited and asked the question, has the promise of primatology been fulfilled or not? If you fancy a read, but I hope that I've shown you, I wanted this talk a little bit to be part about the value of integrative biology and part a kind of love letter to long-running field sites and the kinds of value that you can get if you can get a really integrative research program at a long-running field site. There are huge rewards and insights that I think there's no other real way of approaching some of these questions. So with that, I'll stop. Thank you very much for listening.
All right, James, thank you so much for that delightful talk. And I knew that your work would have something for everyone in it. And I think that you've definitely fulfilled on that point. So right now, what we normally do is just allow folks who are here in attendance to raise their hands. And if you have a view on the right-hand side of your screen, perhaps you might see the participant list and folks who have their hands up. Is that accessible to you? If it's not, I can just decide who to call on. So the first hand that I saw go up, I believe, was Katrina. Katrina,
yes,
would you like to?
I see a few now.
Okay.
Yes, thanks. I was trying to dig across the screens.
Understood. No problem.
So thank you for a really, really interesting talk. I think it did came across as a love letter to primatology, I would say, and I really enjoyed it. And there were so many good ideas about interaction between social knowledge and communication, and the findings about the aging and the changes in social interaction are very interesting. And I really like how you phrased it, how social environment actually gets under our and animal skin. But what I wanted to ask, or maybe it's more like an observation, and I want to make sure that I actually understood or maybe misunderstood what you said at the very beginning of your talk when you were framing your talk. I like how you said that, we can think of it as a mutual made choice. But then you also mentioned that in primates, it's thought that male-male competition is sort of like a driving force in primate sexual selection. But then when I was watching your talk, and I wonder maybe I misunderstood, I was thinking, well, but maybe both sexes are just sort of solving different problems. So, you could think very simplified way, females are looking for access for food, there's offspring care, or maybe, in Cayo Santiago, they're picking mate's face, right, for whatever reason. And then males are, solving a problem of, simplified sort of access to mates. So maybe it's not that clear that male-male competition is a driving force, but it's just like this, the different problems are being solved. And maybe one of them is driving, but maybe it's not as obvious to me, at least after watching your talk. So I just wonder what you think about it.
Yes, it's a great question, and it gets to the heart of something that I've sort of glossed over, and so it's great to be able to talk about it a bit more, which is that is the classic story about primates, right? It's male-male competition. They're like the other large terrestrial mammals, the alpha males, and he monopolizes all the reproduction. And the new male shows up and attacks the alpha male. And if he wins, he kills the infants and takes over the group. And to be fair, that is a lot of what we see in these polygyandrous mating systems, but it's not what we see at this end of the continuum nearly as much. And so there's a lot of primate species that are over here where these crested macaques are. They have strong direct competition, and the difference is really about female synchrony. So at the field site in Tankoko, where we collected the data on the crested macaques, they're highly asynchronous. Females are almost never overlapping in their fertile phases. They reproduce whenever they come back into cycling, etc. And so an alpha male just picks off every female fertile phase. He just mate guards them. And then, of course, there's very little scope for direct female mate choice. There's strong, look at the size of these canines. I mean, it's incredible. There's really strong direct male-male competition, really strong selection on body and canine size dimorphism. The females are much smaller. Look at the testes. There's no indirect competition at all. The testes are absolutely tiny, no sperm competition. And this is the kind of dynamic that a lot of these primate systems are in. And I would say it's spectacular. It can produce really spectacular phenotypes, and people get a bit preoccupied by it but it's not that interesting, actually. It's like, who gets all the, we have a study and we show that the alpha male gets 90% of the sires, right? And it's just kind of like, it's not that interesting. It's like the fitness landscape is very steep and kind of unidirectional. And it's like males are just getting bigger and more armored, and then they fight, and then they win, and then they monopolize the females and they get a reproduction. And it's just kind of like, and that's what's happened in mandrills and drills and gelada and other highly sexually dimorphic primate species too. And it produces spectacular phenotypes like a mandrill. You look at a mandrill, you're like, wow, it's incredible. And then over here, you've got these little drab things like Rhesus macaques. And from just a cursory look, you would think they're not that interesting from a sexual selection point of view, because they're drab and they're not so colorful and they're not so dimorphic and all this kind of stuff. But actually, it's the really interesting end, because what's happened there is they've combined very large group sizes with seasonality and reproductive seasonality in their natural environment. And the upshot of that is high female fertile phase synchrony. And so loads of females are fertile at the same time. The male reproductive skew comes down and down and down. And then the difference between being 5th ranked, 4th ranked, 3rd ranked, they don't barely fight over it, if ever. They have a succession they join the group at the bottom and they rise via succession over time. And the dimorphism is low. And then they have these massive testes volume. They still have some direct contest competition. There's massive testes volume. Faces that are evolving to be ornaments that are chosen in mutual mate choice and all this kind of like a super complicated fitness landscape with like males and females choosing partners, some direct competition, some indirect competition, endurance rivalry, sperm competition, some direct fights and stuff. It's a super interesting fitness landscape with loads of things going on, and it's rare, I would say, for a large terrestrial mammal actually to be like this. It's more of like something you see in birds where you get both the scramble competition and lots of sperm competition and colorful ornaments and things. And to see that mix of traits, I just think it makes them such an interesting species. And you wouldn't, I think like intuitively you would just think, oh, I want to study selection, I'm going to study mandrills or something, but actually right. I think if you're interested in these kinds of like complicated fitness landscapes and like a real mix of sexual selection pressures and dynamics, you've got go and find these like drab things, and then you'll see that there's just so much going on. So yeah, I agree with you, and I'm sorry that I didn't unpack it very well, but I agree with you, there's loads going on in Rhesus. So cool.
Thank you so much. Yeah, this was really, really interesting. Thank you.
Thank you.
Okay, next question, Joe.
Thanks, James. Magnificent talk.
Thank you. Thanks, Joe.
Remarkable setup. It's not just, sounds like it's not just one research program, it's several
lifetimes.
Yeah.
Yeah. Well, I mean, it's built on the back of people who spent time there in the past and kept it going, like yourself.
Well, thanks. So my question is about the redness and darkness of the face as something that, so what's the latest on what kind of quality that those might be signaling, if anything?
Yeah, it's interesting, isn't it? I mean, you would think it's a signal that's easy to link to quality because it's about blood flow and degree of oxygenated blood. So what we think is that the luminance is really about blood flow, and like high levels of blood flow like highly saturate the face and make it like quite dark, and that the redness, the chromatic component, is really about how oxygenated to deoxygenated the blood is. And you could be potentially interested in both of those. Well, not interested in them, but you could get selection for mate choice for them because the resulting offspring, you're more likely to conceive and the resulting offspring might be a better quality. And so I think, this result, we're showing that females that have redder, with lots of variation because there's a lot going on, but females that have redder faces have had higher fecundity. So it could just be related to their underlying condition and their health, and they're healthier and they have red faces. And then as long as males are choosing and using that in their mate choice, they're going to end up doing better. And soon the selection for the preference will spread because individuals that are preferring redder faced females are mating with females who have higher annual fecundity of infants. This is fecundity of infants that survive to at least 12 months. But I think that is generally a huge area that we need to dig into. Really what is being signaled, or at least what's the relationship between variation in the trait and lots of underlying measures of physiology and health and biology. We have those data. So I think, we're in a great position to make this one of the best understood sexually selected traits, actually, because we can do so much.
Well, thank you. So I think about, the classic Hamilton and Zook idea of ornaments signaling resistance to pathogens and parasites. And so has this been connected in any way with the challenge that these animals have with pathogens or parasites?
I don't think so. There's a bit of work on oxidative stress, but I agree with you. I think that essentially that whole area of what is being signaled and why they prefer it is the big black box, really. Sorry, I don't have good answers for you, but
Yeah.
it's an excellent question.
All right. Thank you.
Thank you.
Sorry, if I could just jump in here as kind of a follow-up, because to Joe's question. So with studies, like even Darwin's writing about coat coloration in cats and correlated characters. And we know, for example, that melanocytes, which are involved in production of pigment, have multitudes of effects in the body in general. So there's a story here about genetic pleiotropy. Do we have any information about if there's any role for genetic variation in impacting degrees of redness? And if those genes have been discovered, what other function do they play in physiology that could be involved?
So the interesting thing is that it's actually very plastic. So if you shave the skin next to this red signal, the skin is white, of course, under the skin. It's not bright red. And the difference is if you take a little skin biopsy from the adjacent skin, and then you take a skin biopsy from the face skin or the rump skin, it's about estrogen receptor expression within the skin. So estrogen travels around the body, and then it binds to estrogen receptors targeted in the skin, and it causes a reddening. So the mechanism is not that complicated, although there seems to be this disjunction in males, like that testosterone travels to the skin. In males, it's aromatized one step to estrogen, it binds to the estrogen receptors and has the same effect. But as I said, there's this sort of decoupling of it in males. You can inject them with testosterone, they go bright red. But naturally, there does not seem to be a naturally recurring relationship between circulating testosterone and the redness. So we're starting to having to dig into the changes to the tissue-specific gene expression and try and take biopsies and look up and dig into what's going on. The variation between individuals, I think, as far as we know, is just related to the amount of is plastically related to the amount of estrogen receptor expression, rather than differences in like SNPs at a loci or allelic variation or anything like that. But I think that there's a lot still to be done. And I think the questions are also making me think that I shouldn't have tried to claim that we understand this signal so well.
No, you didn't give that impression. I was just curious about whether there were some genetic variations that have been identified. It sounds like that isn't the case, so it's very helpful.
Thank you.
Jeff Steibel.
Great, thank you, James, and absolutely brilliant talk with a lot to unpack in an hour. So you're a quick talker, and what an amazing research playground in a controlled environment. I mean, incredibly lucky to be able to study there.
Yeah, I really am. Yeah, it's a real gold mine.
I'm going to keep it to a single question, although I do have more. I suspect others will as well. But I'm actually curious whether the social dynamics changed after the hurricane, and whether you saw any phenotypical or genotype changes in some of those more isolated individuals.
So what we showed via in the analysis that Camille led, that's in the current biology paper, is that, okay, so firstly, we see these much more dense proximity and grooming networks. But to be fair, that could just be a relatively simple effect, which is that most of the shade on the island was destroyed. There's most of the vegetation and most of the buildings. And so then all the monkeys are clustered in the remaining bits of shade. But there's a bit more going on to it than that, in the sense that the individuals that were socially isolated before in the behavioral data were the ones that were doing more of the reaching it out and making and building new social connections. Now, of course, you could argue, well, they were the ones that were isolated in the first place, and so they're more likely once they cluster together. I guess another question I saw that sort of crops up to me when I think about this is like, does it matter if that's the mechanism? If the mechanism is just very simple, like, oh, in the response to natural disaster, they all have to cluster in a particular area and then they all become new friends and build new social relationships. That's just the mechanism. Like the effect, of course, is that we see this big change in their sociality. And it's interesting actually, because I know some people that ended up in hurricane shelters for a while after Hurricane Maria, and they're still friends with the people that they were sort of forced to densely live with in these hurricane shelters for like 10 days after the hurricane. And so it's you know, the mechanism isn't necessarily it doesn't really matter. It's had the social effects, right?
Yeah, and in particular, if you look at human studies, you see a lot of health benefits to that interactivity. Again, even if it's forced, and I was wondering if you're seeing that, you know, on the island as well.
Yeah, right. So is there a health benefit? So we have an R01 from NIA at the moment, which is basically called Social Modifiers of the Pace of Aging. And in that analysis, essentially, all these molecular, physiological, physical health measures are being combined as a sort of dependent variable, where the independent variable is like sociality. And we're just asking kind of like, how does sociality, variation in social environment get under the skin? And that's in like year 3. So that's the plan, to get exactly at this sort of stuff. Like when you end up in these different social networks and with different degrees of social support, how does that end up impacting your health? And of course, really, you can consider all your age is a primary predictor of health. And so to a certain extent, you can consider all health questions to be an aging question. Like, are you biologically young for your chronological age, or are you biologically old for your chronological age? And then what determines variation in that pace of aging?
Good. Look forward to seeing the results.
Yeah, thank you. Me too. I'm extremely excited about it.
Molly?
I'll just echo everyone's sentiment. Thank you for such a wide-ranging, inspiring talk. You really gave us a lot to think about. So I was just wondering if you had any observations about social distancing in the face of infection, or sort of manipulations to the normal social networks or practices. I know that you mentioned this 2010 Shigella epidemic that the macaque community experienced, and so maybe there was some response at that point, and of course the recent work in social distancing among animals, inspired by the human pandemic going around, made me think about this, and you have such rich social data here.
Yeah, that's a really, really good question. I don't think we've yet been able to dig into social avoidance, but we do have a really cool new analysis that's been led by a PhD student of Lauren Brent's, who's one of my co-PIs and collaborators on all of these projects, Melissa Pavez, and she's done this really cool analysis where I think you're probably aware that there's a lot of data, including from primates, showing this relationship between sociality and fitness. But without a lot of data on the mechanisms of what's linking sociality and fitness. And we can show that on Cayo, one of the sources of deaths come from woundings. You get a wound and it leads to death, and we can show that. Individuals that get wounded have a higher risk of mortality. And we can also show that individuals who are better socially integrated have lower wounding risk. So we actually have a mechanism on Cayo, at least one mechanism. I'm sure there's plenty and they're not mutually exclusive, but we have a clear mechanism in that individuals that are better socially integrated have lower wounding risks, and higher wounding risk leads to higher mortality. So I think we're starting to get there, where we're like picking apart the links between diseases, infections, wounds and things and the benefits of sociality and what that looks like. We haven't looked at your specific question, which I think is a really particularly interesting one, about whether you see things like avoidance of individuals that have infectious diseases.
Is the wounding, is that like conspecific conflict, or
it's
is that
mainly conspecific conflicts? They also sometimes just have accidents, but it's mainly conspecific conflicts. Yeah.
very interesting?
I don't see any other questions, but I thought, given that opportunity, I'd ask you about the elder females and the period of time that they're in a post-reproductive state. What is, I obviously need to read the great articles that you showed the figures on with Lee et al. But is there common observations of post-reproductive survival among females in this population?
So there's no real post-reproductive survival. What seems to happen is that they keep reproducing, the interbirth intervals extend, and they get to the point when they're like in their mid-20s where the infant mortality is 100% or close to 100%. So they're old. And I mean, they really look old. I was just on, I only flew back from Puerto Rico last night. So like, I was been there very recently, and I took video of some of these individuals walking. And, you know, the old individuals can really, their hips are gone, their back, the vertebrae in their back are fused. They're staggering around. They look dreadful. They do not look like well individuals, and their interbirth intervals are long. They still reproduce with basically 100% mortality. So they don't seem to have like a they're post-successful reproduction, but not post-reproduction.
Okay, thanks for that point. There's a question by Katrina.
If nobody else want to ask a question, because I already did.
I think you're the only one. This is your time.
Great. It's my time. So I actually want to kind of pick on something that you mentioned and see if you want to elaborate a little bit more. So I also thought a little bit about the relationship between sociality and fitness. And it was really interesting, you know, that you mentioned you seem to find this one particular mechanism of social integration and reduced wounding, which I also agree that I think that's, you know, that's the missing point right now in primatology, to actually, you know, figure out what are the mechanisms.
Yep.
And the daunting sort of thought for me is that it might be a little bit hard to find, you know, I imagine there are a lot of things going on. Like there's not one mechanism,
yep.
Right? So yeah, so I wonder just how you think about it, because I was kind of worried to chase one particular mechanism and then you don't find it. What does that mean? Does it mean that, you know, so how do you sort of think about it? Thank you.
Yeah, I think that's a really, really interesting question. I mean, we have clear relationships between sociality and fitness on Cayo, like a lot of people do in these long-term data sets. I'm hopeful that the current studies that we're doing, where we're tracking variation in the social environment and looking at all these different kinds of health, molecular, physiological, physical measures, we can start to unpick this a bit. I think that, you know, in the social aging review that we just had out, which I can try to go to this paper, you know, there's some really interesting literature, I think, about changes in mobility, changes in sensory systems and sensory decline, cognitive decline in the human literature. Which way round is this? Is the question, right? Are individuals losing their mobility and their ability to hear people and see people and things, so they stay home because they no longer find going out and about and having social relationships to be very rewarding? Or is their social lives deteriorating and it's impacting their health? Or is it a loop and a feedback loop, and it's all happening at the same time? But we're seeing, you know, in our environment, in our studies, we're seeing decline in sensory systems like vision over time, decline in hip mobility and kinetics and your ability to get around, and then social selectivity with them focusing in on special long-term friends and long-term partners and sacrificing the relationships and stuff. And we explore in this review some of the ideas because, of course, you know, it's common for my parents, for example, will forsake seeing their friends to see their grandkids now. So and of course that could be adaptive because they're investing in the time that they have left, and the investment they have left to give is going into individuals with our values. So you could imagine that as demographics and change, and you end up surrounded by your own descendants as you get older, and you don't have much investment left to give, you might choose to give that investment into them. So I think that let's come back to your question more directly. I think that it's really hard to identify those mechanisms. I'm hopeful that by having these rich social data sets and lots of other kinds of data types, we're going to be able to dig into what some of those mechanisms are. I think that the excellent wounding analysis that Melissa has done is a starting point for us. And I really hope that we're going to be able to dig into more of these, like this sociality, health, fitness axis as it is, right? Yeah.
Thank you. Thank you.
Thank you.
Question from Dan.
Hi, James. I'll just second what everybody else has said about an amazing, total compendium of remarkable results. I want to focus in on one particular thing that's come up in the discussion, and I several times actually. And forgive me if I know you published on this probably 10 years ago or more, and I haven't revisited the paper, so I may be getting the details wrong. But as I recall, although my colleagues and I found no support for the terminal investment hypothesis in chimps and humans, you and your collaborators did find some support for it on Cayo.
A little bit.
Yeah, a little bit. And so my question is, look, if neonatal mortality is 100%, then birth weight's just the wrong variable, and you're really not seeing, you're just seeing kind of a broken system breaking down further. You're not seeing an adaptive allocation of resources as a function of reproductive potential, right?
So what we do see in those older females, and that's the Hoffman et al. 2010 paper you're talking about, right, in behavioral ecology, is there are some behavioral data in that paper as well, and we do show that as individuals get older, they allow the infants more access to the nipple as they age. Actually, it's cross-sectional, I should say. I'm sorry, it's not a longitudinal analysis. So I do think there's bits of evidence like that. Like, yes, they're having worse outcomes, but they are providing, we don't know about the actual milk, right? But they're at least providing more contact, allowing more ventral contact and more nipple access. You could say that that's some evidence for attempts at terminal investment. It's not strong evidence. I think there's very strong evidence of senescence in this population. You can see it across all of the different kinds of measures that I'm showing, and little bits of evidence for terminal investment. But I agree with you, there's not a lot of strong evidence for terminal investment.
Yeah, I mean, it really looks in a lot of ways like they're just kind of constraints on optimality, right? Because if the system was highly optimized, reproduction would cease sooner with more positive outcomes, and potentially, life would end correspondingly, right? And this kind of slow running down of everything just looks like maybe selection is weak at that end of the lifespan. And,
or at least it is on Cayo.
Yeah, that's
maybe it's much stronger in the wild because they just get taken out by predators once they get
good point.
20 or whatever. But on Cayo, it's relaxed. There's no predators and they keep getting fed. And so I think probably there's weak late life selection, and so some of those. But it's remarkable because we'll submit a grant and we'll get told by reviewers at NIH that our animals are unnaturally short-lived. because because they're all working in captivity where they can keep them till 30 years old in a cage on their own, just like literally spoon feeding them, and they're blind and they can't move and they're obese or whatever, and they're saying, you don't have many individuals over 20 in your population. Like you have to justify towards the short-lived nature of your population, unlike the natural models that we have where we can keep them alive till 30. And it blows my mind that people would think like that. Of course, actually, it's the other way around. If anything, we have an unnaturally large number of old individuals, actually, probably.
Yeah, well, reviewers are, of course, humans with culture, and so they have their own norms. But I mean, our data, arguably, were all zoo-fed populations because they were captive chimps and weird humans, right? And in both of those, we don't see evidence where essentially there's no predation and there's super abundant calories.
Yeah.
Still, it's not there. Yeah.
Terminal investment, you mean?
Yeah, that's right.
Yeah. I would say we haven't. Now we have much bigger data sets. We could probably revisit that question. Actually, I think it's an important one to revisit. I think that in that original Hoffman paper from Christie's PhD dissertation, there were little bits of evidence like that, like the behavioral stuff that old females might have been increasing the investment that they had, but certainly the outcomes were still worse, as you say.